Your eyesight isn't getting worse because your eyes are wearing out. That's the intuitive version, and it's wrong in a way that actually matters. Vision loss with age is a metabolic event happening in tissue so specialized it can't replace its own proteins. What you feel as blurs is structural damage to molecules that have been in your eye without repair since before you took your first breath. Stop on that for a moment, because it changes what we're talking about. The proteins that give your lens its transparency, the ones responsible for bending light precisely enough that you can read these words, formed in utero. They have a half-life exceeding 70 years with effectively zero turnover in the lens nucleus. They're not being swapped out, not degraded and replaced. They are, in the most literal sense, the same proteins you were born with. Every glucose molecule that's moved through your bloodstream since your first meal has had access to them. And glucose, given time and protein it can't escape, does something very specific. It sticks. The process is called glycation, not to be confused with glycation's more famous cousin, the kind your doctor checks with a blood test. Glycation in the lens is non-enzymatic. Nothing controls it. It's a spontaneous chemical reaction between glucose and the amino groups of proteins, particularly lysine and arginine residues in a family of proteins called crystallins. The reaction runs through a series of intermediate compounds, Schiff bases, amidori products, and eventually produces what researchers call advanced glycation end products, or AGGs. The word for what AGs do to crystalline proteins is cross-linking. The accurate description is welding. Once two crystalline proteins are cross-linked by an AGG, they don't move independently anymore. They're bonded chemically through a linkage that doesn't come undone. And here's what cross-linking does to a lens. It stiffens it. Research published in the Journal of Biological Chemistry in 2020 by Nandi and colleagues found a direct correlation between AG-mediated cross-linking levels and the measurable stiffness of human lenses as they age. The relationship wasn't suggestive. It tracked stiffer lens, more AG cross-links, full stop. Have you ever noticed you're holding your phone slightly further away than you used to? Or that restaurant menus require more light than they once did? That experience, that small, slightly embarrassing recalibration of where reading distance now lives, is the mechanical consequence of what I just described. The lens focuses by changing shape. Tiny muscles around its edge pull on it. When they relax, the lens rounds. When they contract, it flattens. It's been doing this continuously since infancy, adjusting focal length in milliseconds. But a stiffened lens doesn't reshape easily. It resists. The range of focus narrows. By 40, almost everyone notices it. By 45, nearly 90% of adults have measurable presbyopia. By 50, it's essentially universal. Tell me in the comments, do you already reach for reading glasses, or are you still pretending the print is just getting smaller? Yes or no? The responses from people who swear the menus have changed are always the most entertaining. So the reading glasses aren't really an eye problem. They're a material science problem. Which means the question becomes, what drives the chemistry? Here's where it gets less simple and more interesting. Glucose is the obvious culprit, and it matters, but the lens turns out to be particularly vulnerable to a less obvious glycating agent, ascorbate, vitamin C. The lens accumulates ascorbate at concentrations roughly 20 times higher than in blood plasma. The idea is protective. Ascorbate is an antioxidant, and the lens is constantly assaulted by ultraviolet light. But ascorbate is also a reducing sugar. It degrades. It produces reactive carbonyl compounds, including a glycating agent called 3-deoxythreozone. Research on AGE formation in lens proteins, including work from Nagaraj's lab at the University of Colorado, has identified ascorbate-derived AGEs as significant contributors to cross-linking, operating through a pathway distinct from glucose. The very molecule the lens accumulates to protect itself becomes, over decades, one of the things slowly stiffening it. I find this strange every time I think about it. The defensive mechanism participates in the damage. Not because it fails, because it runs. Long enough, the lens story is only half of this. And the other half doesn't announce itself. No blur, no squinting, no holding things further away. Just a single layer of cells at the back of your eye quietly losing a battle it's been fighting since your 20s. And by the time you know it lost, the damage is permanent. The retina is the most metabolically active tissue in the human body. Otto Warburg confirmed this in the 1920s. The retina consumed more oxygen per gram than any other normal tissue he measured, in the same range as some of the most energy-hungry cells in biology. Behind the photoreceptors, the rods and cones that detect light, sits a single cell-thick layer called the retinal pigment epithelium, or RPE. 1 cell thick, the width roughly of a soap bubble. And it is simultaneously phagocytosing shed photoreceptor debris, transporting nutrients from the blood supply, recycling visual pigments, and managing the metabolic waste of tissue running at maximum intensity. Every hour, every day, without a break from the moment your eyes formed. The outer retina converts more than 80% of the glucose it consumes directly into lactate. The RPE absorbs that lactate and runs on it. Think of it as a metabolic service station wedged between a Formula One engine and its only fuel supply. The engine never stops. The station never closes. And the station is 1 cell thick. Here's what nobody tells you about AMD. It isn't a disease of old age. It's a disease with a visible endpoint in old age. The accumulation that causes it starts decades earlier. Beginning early in life, not your 50s, not your 40s. The RPE gradually accumulates what researchers call lipofuscin, molecular debris from the incomplete digestion of shed photoreceptor outer segments. A healthy young RPE clears this efficiently. As it ages, clearance slows. Lipofuscin builds inside the cells. It's photoreactive. Visible light hitting lipofuscin generates reactive oxygen species, which damage mitochondria, which reduces the cell's capacity to manage oxidative stress, which produces more damage. It's a slow compounding loop with no obvious symptoms running in tissue you'll never feel for decades. What eventually becomes visible as AMD starts as drusen, deposits of lipid and protein waste accumulating between the RPE and its blood supply, like sediment building between a filter and a pipe. Small hard drusen are a normal feature of aging. Soft, large drusen are the early warning. They signals that the RPE is losing the battle with its own waste, that the blood supply is being compromised, that the retina is beginning to starve. When enough RPE cells die in what's called geographic atrophy, the photoreceptors above them die with them. The loss of central vision in AMD isn't a sudden event. It's the end of a process that's been measurable, had you known to look, for decades. The gap between what that sentence means and what most people know about their own eyes is the thing that pulled me into this work in the first place. Not the science itself, the gap. It felt like something worth doing something about. So here's the uncomfortable question the mechanism raises. If this process starts decades before vision changes, and the tissue involved can't repair itself, what can actually be done? The retina, it turns out, built its own answer into the architecture. The central region, the macula, contains a layer of yellow pigment made up of 3 carotenoids, lutein, zeaxanthin, and mesozeaxanthin, measurable as macular pigment optical density, MPOD. It does 2 things. It filters short wavelength blue light before it reaches the photoreceptors, and it acts as a local antioxidant, neutralizing the reactive oxygen species that the retina's extraordinary metabolic rate continuously generates. The retina doesn't synthesize these compounds. It can't. They're entirely from diet, which means macular pigment density is not fixed, it's responsive. It's built from what you eat. A 2021 meta-analysis covering 46 studies and over 3,000 participants found that MPOD increases measurably at intakes of 5 milligrams of lutein per day or above. The ARIDS-2 trial, over 4,000 participants, 10 years of follow-up, funded by the NIH, found that 10 milligrams of lutein with 2 milligrams of zeaxanthin daily reduced AMD progression by approximately 26%. That number belongs to people already at high risk, intermediate AMD or advanced AMD in one eye. It wasn't a prevention trial in healthy eyes. Worth being precise about that, because a 26% figure on a supplement label doesn't always come with that context. What the mechanism tells everyone, regardless of risk, is what the retina was built to receive. The tissue has dedicated transport proteins specifically to concentrate lutein and zeaxanthin there. It built the infrastructure for these compounds. The average Western diet delivers typically under 2 milligrams per day. 5 milligrams is a large plate of spinach. The gap between what the retina is getting and what it was designed to run on isn't a pharmaceutical gap. It's a grocery gap. And then there's the exercise finding, which is the one I didn't expect when I first read it. A 2018 study published in Metabolism, Clinical and Experimental, examined the effects of 4 weeks of high-intensity interval training on retinal perfusion in healthy adults using optical coherence tomography angiography, a technique that images retinal blood vessels without contrast dye. 58 participants, hit twice weekly. Measurable changes in macular perfusion after 4 weeks. Separate research in type 1 diabetes patients without clinical retinopathy found large improvements in retinal microvascular parameters after the same duration. These are small studies in specific populations, and the direct long-term implications for AMD prevention in healthy people haven't been established in large trials. But the mechanism is well supported. Improved cardiovascular fitness improves microvascular health, including in the retina. Decreased choroidal and retinal blood flow is a documented risk factor for AMD. The retina is not exempt from the vascular benefits of exercise. The magnitude in healthy aging populations isn't yet quantified with precision, but the direction is clear, and 4 weeks is a short runway. The lens is a harder target. There's no dietary equivalent of lutein for crystalline cross-linking, no compound with the same mechanistic fit and evidentiary weight. What the glycation chemistry points toward is less specific and more uncomfortable. The lens is experiencing whatever your blood glucose is doing, continuously, without the ability to undo any of it. Diabetic cataracts are the fast version of this story, the same Ag-driven cross-linking that causes age-related presbyopia and cataracts, compressed into years rather than decades by sustained high glucose. The mechanism doesn't respond to a supplement. It responds to the total glycemic burden the crystallines have been exposed to across a lifetime. That's not a satisfying answer. It's just the accurate one. And here's the honest gap, which I'd rather name than skip over. There is no pharmacological agent that currently reverses established Ag cross-links in human lens tissue. Research from Nagaraj's lab at the University of Colorado, published in 2020 and 2021, identified compounds that inhibit cross-link formation in animal models, trapping reactive intermediates before they weld proteins together. The animal data is genuinely interesting. Human trials haven't happened yet. Presbyopia reversal is being worked on. But being worked on and available are different sentences, and right now only one of them is true. So here's what the full mechanism, taken together, actually tells you. Your lens proteins formed before you were born and will never be replaced. The damage to them is cumulative, invisible, and chemically irreversible once it's done. Your RPE has been accumulating waste since you were young, running a metabolic operation of extraordinary intensity on a one-cell thick platform, and the failures it accumulates are permanent. None of this is fixable after the fact. All of it is influenced by what happens before. The retina was built to concentrate specific dietary compounds it can't synthesize, and most people aren't delivering them. The retinal vasculature responds to cardiovascular fitness with measurable changes in 4 weeks. The lens crystallines are exposed to whatever metabolic state you sustain over decades. The actions the mechanism points toward aren't new. They're not a protocol. They're the same things that keep every other high metabolic rate system in the body running. Except now you know which specific tissue is paying the price when they don't happen, and exactly how it's paying it. What you noticed in the blur of a restaurant menu, or the phone held slightly further away than last year, is not the beginning of this story. It's the middle. The beginning was quiet, invisible, and decades earlier, in tissue doing chemistry that has no symptoms, running the slow arithmetic of damage you're only now reading the result of. Understanding that doesn't change what's already happened. But it's the only thing that changes what happens next.